Okay, just want to remind you again, if you have a Wi-Fi device, please turn off your Wi-Fi signal so the recording isn't interrupted by something like that. I want to introduce you today to Dr. Sauter, Dr. Ed Sauter. He is the director, not just somebody who works over at the Cancer Research, but the director of cancer research at the what we used to call the UT Health Center down the street by Tyler. And that has a similar history to Laterno in that it was also an Army hospital of some sort, and maybe he'll tell us about that. But Dr. Sauter has both a PhD and an MD. So he did a PhD, MD program. And so he's both a practicing medical doctor and he's a researcher in cancer treatment, which is what they do down the road over there. And so I hope that you will realize that this guy is a big name in cancer research and appreciate what he tells you about what's going on here, and that you will also take the opportunity to join us at lunch after the seminar. We have a couple students who are brave enough to join us, but if you have any interest in this topic, I hope that you will join us there and continue the conversation with Dr. Sauter at that time. But until then, let's give him a warm welcome.
Well, thanks again, Dr. DeBoer, and everyone else for having me. So the first thing I want to know is this is what is this a wasp or what is this? A yellow jacket, okay. But it's got blue. I think it's got some yellow as well. Anyway, so yes, I do both clinical work and also research. My area of research is breast cancer, and that's what we're going to talk to you about today. Specifically, one area of breast cancer that you may not know so much about. How many people have a family member, relative with breast cancer, history of breast cancer? Okay, we're talking like at least a third up to a half of the audience. Big deal. It's the most common cancer in women, and it's on the increase in incidence, on the increase. Not in mortality, fortunately, because we're working hard to cure it, but it's on the uprise in incidence. So one of the things that we don't understand about breast cancer is still really is how it forms. And we really do not understand how breast cancer forms in young women. So what causes breast cancer? Any ideas? Why do women get it and not men? Men can get it, but only 1/100th as often. So why do women get it 100 times more often? Well, there's probably more breast volume, of course, but that's not the only reason. Hormones, right? Estrogen. Estrogen is inducive or permissive for the development of breast cancer. It doesn't necessarily cause breast cancer, but high hormone levels make cells make mistakes. And normal cells start dividing. And during those normal cell divisions, mutations occur, and other protein and RNA changes, and some of those drive the development of breast cancer. And hormonally regulated and driven breast cancer is the most common cause, but not true in young women nearly as often. And in fact, for cancer in general, what do we know about the younger a person gets it? The more common it's genetically driven. That is, inherited. Or there's just a unique mutation which occurred rather than some hormonal influence which causes the cells really to just rapidly divide. In any case, we're going to talk about pregnancy-associated breast cancer. Let's see here. I'll figure this out sooner or later. So I just want to go over the anatomy of the breast. These are ducts. are tubes. So the breast, of course, enlarges under the increasing levels of estrogen and progesterone that occur during the time of menarche in a young woman. And these tubes that are very rudimentary in a man lengthen as the breast enlarges. But there's these little grape-like clusters called lobules, and the grape-like clusters are where milk will form under the influence of certain hormones that occur with a letdown process when the baby starts sucking on the nipple. And so really, those lactogenic hormones are what cause the milk production within the breast. So the breast enlarges initially, of course, under the influence of estrogen and progesterone, which levels are high when a woman's pregnant. But the formation of milk actually really only occurs under the stimulation of suckling of the baby, and that's when the milk forms. But what happens? Think about it during when a young woman or an older woman gets pregnant. The breast enlarges, right? More glands are formed, more tubes form, more ducts form, more stuff happens. How does cancer form? Anybody know? It forms when cells make a mistake. Does cancer form when cells divide or when they're dormant? When they divide. A dormant cell can make a mistake. It's not doing anything. So the more rapid that a cell divides, the more likely a cancer is to occur. Even if something's not driven to make a mistake, just random errors. When you learned about DNA, right? It divides in two and then doubles. During that process of splitting apart and then duplicating itself, there's random mutations, about 1 per million. And when you got 3 billion bases, there's a frequent chance for mutational events to occur. So the more that a cell divides, the more mutations occur, the more likelihood there is for cancer. Where do cancers occur in a person's body? You know, you all have cancer cells in your body right now? All of you. And me too. It's just that our immune system is fighting them so they don't divide. Well, this is a perfect setup during pregnancy and lactation for growth, cell division, mutational events, and cancer. Second problem, it's hard for a woman to feel the lump because the breast is hard, full of milk, and enlarged. So it's a perfect setup for a real problem. We'll go over a little bit of history here in detail. So what is associated with this pregnancy-associated breast cancer? We know that, first, pregnancy influences breast cancer risk. Now, I shouldn't have shown you this slide, but I'm not going to show you that yet. Does pregnancy increase a woman's risk of breast cancer long-term or decrease it? Does it increase or decrease her short-term risk? Actually, that's pretty close to correct. You know, the party line is that having a baby decreases your risk for a woman's risk of developing breast cancer. Well, that's never true in the short term. Risk always goes up. I mean, it has to. The breast is dividing. Mistakes are occurring. If the cells don't die and they're driven to cancer, you can have more cancers. So short-term risk for women is always higher with pregnancy, with or without nursing. The long-term risk depends upon when you have your first child. We can discuss that, but that's another talk. It would take too long. In any event, age at first full-term pregnancy. Early age leads to permanent changes that combat abnormal cell growth, and basically that's called terminal differentiation, where the cells cannot go back to like a stem cell. And stem cells in cancer are very close
to each other. In fact, there's this whole idea that cancer stem cells exist. It's a debatable argument issue, but if cells cannot revert back to a stem-like environment, then they can't really deform cancer. The other things that influence risk are parity, how many babies you have. That's been proposed anyway. More babies, lower risk. Whether that's accurate or not is debatable. The length of lactation, the longer you nurse a baby, the less likely a woman is to get breast cancer. That's also in question in family history of breast cancer. So in any event, these are things that have been claimed to be true, and again, I am not anti-children nor anti-nursing. I think it's a great thing, but I think that it needs to be understood that it's not like it's going to help you in the short term in terms of the woman. I mean, when she nurses her baby, she has to be aware that it does increase her risk of breast cancer, at least to some degree. And if she's an older, in her 30s when she has her first child, then it increases it even more. Yeah.
What is the first child, the age that your first child have to do with it?
That's when so when the breast enlarges at the end of nursing, it goes back to normal. At the end of weaning, it undergoes what we call breast involution. And actually that breast involution is just as important as the enlargement. In other words, it's basically you build a house and then you tear it down. But that tearing it down, anybody knows how the body tears itself down? Matrix metalloproteinases. You ever heard of those? MMPs. Matrix metalloproteinases. It breaks down the extracellular matrix, and basically the epithelial cells fall apart when they don't have the matrix to hold them together. So when the MMPs occur, growth factor upregulation occurs, cancers form there too. But if there's the potential for cancers to form. If cancers don't form, that process of that the cells enlarge and then shut down in the breast leads to what we call terminal differentiation, where the cells are less likely to become stem-like or have the chance to become more like cancer with the next pregnancy. So that first pregnancy is the key one. Okay. Second, third, fourth, the importance is there is some influence probably, but not nearly so much as the first. So this was all interesting stuff, but no one really had taken the next step because the question is, how can you study the influence of pregnancy and lactation on breast cancer in a nursing mother? I mean, it's like hands off. You don't do anything. No, nothing. I mean, even if you feel a lump in the breast, you usually, as a surgeon, say, oh my goodness, we're going to wait until it's all done, and then we'll deal with it, for all sorts of reasons. You mess up the nursing. If you do a breast biopsy during nursing, you see milk leaking out of your biopsy cavity. I mean, it's just, it doesn't heal right, it gets infected. A million reasons why you really just don't want to go there if you don't have to. But unfortunately, occasionally there might be cancer there, and you don't want to miss that either. So, in any case, it had not been well studied. So our idea was to collect milk samples to figure out what was going on and how differences between women based upon proteins in milk collected over time could tell us. And so we collected milk right at the beginning of nursing. 2 months into it, when what we call whole milk occurs, which is when the maximum production sort of occurs. And then during weaning, when the woman takes the baby off the breast, and essentially the breast has to start deconstructing itself, upregulating certain proteins, including metalloproteinases, but not only. Have you guys ever heard of transforming growth factor? beta? Pleiotropic hormone? No. Talk about it in a minute. All right. So I've collected samples from all over the world, both at the institutions I've been in, and I have a Army. You've heard Avon? You've ever heard Avon Cosmetics? Well, they have a special interest in pregnancy-associated breast cancer, and through them I get milk samples from women all over the world, mostly in the U.S. and Canada. And anyway, we've got 300 samples from 300 women, sequential, beginning of nursing, 2 months into it, and when the baby's weaning. That weaning one is hard because the women forget about it. Sometimes they've been nursing their baby for years, and but we've done fairly well. And we've just tried to figure out what's going on in the breast with these changes over time to see if we could predict why, pregnancy and lactation increases risk. And so these are some various processes for studying this. And the first thing we wanted to do was to look what about total protein. And the truth is that the total protein is highest at the beginning, second highest at the end, and lowest at the middle. Any idea why that might be? We won't talk about why it might matter. Okay, yeah.
I remember with it's to give the baby a stuff, kind of.
It might be. The most important reason, more than likely, is because the milk volume goes up. The woman makes more milk once it's sort of geared up. The breast is geared up to make milk. And so initially there's a lower volume, it goes up, and then during weaning it goes back down again. And so there the concentration of proteins, of course, is diluted more with larger volume. So it's really, more than anything else, probably just a dilutional effect of greater volume. All right, a change of topic here. We had access to a unique cohort of samples here, and I'm going to just go over this with you. The question I had related to pregnancy-associated breast cancer was, how do we get at the differences in the breasts, women whose breasts had developed cancer during this time as opposed to women any other time? And I've been studying breast cancer now since 1991, okay, a long time. Maybe, I don't know, some of you may not have been born then for all I know, but anyway, that's how long I've been studying breast cancer. And there's a variety of changes that occur with breast cancer, and 5 of the proteins that are altered are TGF-beta-1, transforming growth factor beta-1. It's a pleiotropic hormone, which basically means when this protein goes up, it can either increase the risk of cancer or it can decrease it. And it depends upon the milieu in the body. Sometimes when it goes up, it shuts down cells. Other times, it makes them grow. So it's a bizarre hormone, and the environment in which it exists makes all the difference. So it's very confusing. Same for TGF-beta-2, very similar. Basic fibroblast growth factor, YKL-40, NGAL, it's lipokaolin. It's neutrophil gelatinase lipokaolin. Anyway, these 5 are well associated with breast cancer. What we had here is we had breast milk from these women who developed breast cancer, were diagnosed during nursing, and we got milk from both breasts, including the one with cancer. Now, breast cancer during this time is very uncommon in young women. So it was a unique cohort, but we were able to match breast, the normal breast and the cancer-containing breast. And you'll see here the mean values for most of these proteins is higher in the cancer-containing breasts. It's TGF-beta-1's an exception, but the really highest ones were in beta-2 and BFGF. So those are the ones that we really got interested in. And this is just an explanation. But of the 5 women, this is the third line, I've told you about the first 2. we actually, 4 women were diagnosed while they were nursing their baby. And again, the challenge was, it went to the doctor, I feel alone. And the whole breast is so lumpy, it's hard to feel anything. You don't want to do mammograms on a woman because of radiation. Basically mammograms are useless anyway because the breast is so dense, it's just a whiteout. So it's a real challenge as to how to image a tumor in a nursing mother. So they did an ultrasound and found it that way. But 4 women were diagnosed during lactation, and 1 was diagnosed 2 years later. And we still had milk from both breasts. And basically, the point was that of these 5 proteins, only 2 were higher in the breast that had cancer in it. And so that was telling us. So you understand the reasoning behind why you look at these things? If something is different in the normal versus the cancer-containing? Because that suggests that this particular protein may drive the development of a tumor. Right? There's a normal level of something in your body, and there's an abnormal level of something in your body. If there's an abnormal level of something in your body, it may be influencing cells to grow, or to become malignant, or to multiply and divide. And so these 2 that were higher, these were abnormally high levels in the cancer-containing breast, whereas the other proteins were not. And so again, this is what we go after. I don't want to get too far off track, but in cancer in general, that's how we figure out what to target. When we take a person's tumor for personalized medicine, what do? We look for mutational changes. We look for changes in protein and RNA. And the things that are abnormal in that person's tumor is what we attack. With something, if it's too high, we knock it down. It's harder if something's low to bring it up, but if it's high, we try to knock it down. And so this is the same strategy. We want to know what's abnormal in that particular tumor. So we went a little bit farther now that my interest was piqued to try to figure out what were the changes in a normal woman's breast that occurs during nursing, and probably try to figure out which of these things we need to focus on most. And again, we had 3 samples: transitional milk, the beginning, mature milk, 2 months, and then wean at the end. So we found there was changes in these kallikreins, and then these 2 others. Remember, we said that this was higher in cancer, and this was higher in cancer. But with normal nursing, this actually went down. So that was suggesting to us that maybe, there was a protective effect of nursing. But again, TGF-beta2 went up, just like it did in the cancer-containing breast. So that was a little bit confusing. We also looked at gene methylation, which is quieting of KLK6. KLK6 is a kallikrein. Kallikreins are tumor suppressors that prevent cancer. And it turned out that this was silencing this tumor suppressor, which is the way that it worked. But we wanted to know, so again, remember that the older a woman is when she has her first child, the greater her risk of getting breast cancer. Why is that? Why is it that if she's 21, the long-term risk goes down, but if she's 35 when she has her first child, it goes up? Well, no one really knows. I mean, there's a hypothesis. But what we found by looking at age at first full-term pregnancy was that there were changes in these same cancer-causing proteins in older women, BFGF, and this other one. And this one here, which we know is a tumor suppressor, went up with younger women. And so these are things that we are going to be looking at some more. So these are the proteins we talked about, KLK6, these 2 tumor suppressors, and TGF-beta2 that went up. And PSA, believe it or not, is made by women too. You ever heard of PSA for prostate cancer? It's actually made in the breast as well. And it goes down in very young women. So I don't want to bombard you with too much, but again, we were trying to figure out how pregnancy and nursing might influence breast cancer risk. And at the end of the day, what do you want to do with this information? You either want to screen women for levels of these things, if they're abnormal, or to target them if they develop cancer. But from my point of view, a prevention strategy or a screening strategy makes even more sense. And again, we'll talk about this in a second. How many blood tests are there to screen for cancer? You know, in women. One, alpha-fetoprotein for liver cancer. That's FDA approved. How many are there in men? Alpha-fetoprotein for liver cancer. Used to be PSA for prostate cancer. That's been pulled. So PSA is no longer recommended for routine screening for prostate cancer. It's another talk about the reasoning behind that. My point is, so that holy grail of screening for people for cancer with a blood test or some other kind of test is hard, but maybe we could screen breast milk for pregnancy-associated breast cancer. And that's kind of where we're going with all this, which protein to look for. Remember, family history increases a woman's risk of cancer too. So we looked at that, and it turns out that a family history may be one of the reasons why not all women are protected when they have a baby, but only the group with a family history had a protective effect from breastfeeding. And again, of these proteins we looked at, TGF-beta2 seems the one that's differentially changed between women with a family history where it doesn't go up versus women without a family history where it does go up. So more and more information is suggesting to us that this particular TGF-beta2 may be where the money is. All right. So we were asking, again, we're trying to figure out how to get at a predictive strategy in nursing mothers for their risk of getting cancer. You know, the women in the audience, most of you get pregnant. Okay, nurse your babies. You might want to know about that, right? You guys, you're going to have a wife. Most of you, same story. It's important. It's coming up, if it hasn't already come to your front door. So, what do you guys know about exosomes?
Cellular organelle.
Okay, so it has been known for a long time that there's little packets of a cell that bud off the cell. So if you have a cell membrane, you have the nucleus, and you have the cytoplasm, and then you have the membrane, correct? But little pieces of the cell membrane bud off all the time. And they contain those little pieces of the membrane that come off have within them, they're kind of mini cells, but they don't have a nucleus. But they have RNA and they have proteins. And in fact, that's a way that a cell sitting here can talk to a cell sitting downstream, like in a breast duct down the stream. So it's a way for cells to communicate that don't touch each other. Obviously, cells that touch each other can directly communicate through cell signaling. But cells at distance from another can also communicate. And one of the main ways is through exosomes. So a little piece of the cell buds off, transports its packet of RNA, proteins, and carbohydrates, and then can enter a cell at distance and communicate. Yeah, and more is being shown that this occurs not only in normal physiologic processes like lactation, but also in breast cancer. So we thought maybe this is one way where, you know, that communication might influence cancer risk. So initially we wanted to see if that hypothesis had any traction. So again, we looked at 3 different milk samples, took it from 13 mothers, extracted exosomes, and then looked at 6 proteins linked to breast cancer. So here's, TGF-beta, a lot of these players are the same. The difference is that most of us in the field think that that last sample, the wean sample, is the most important one because during that involutional process, breaking everything down is when cancers either form or they don't. And in animal models, supports that. Multiple animal models support that hypothesis. That it's at the end, the breast either deconstructs itself and undergoes apoptosis, all the cells, but occasionally some do not, and survive and become malignant. And so, remember what I told you about matrix metalloproteinases? Okay, well we're going to talk about 3 of them, plus milk fat globulin protein, which is important during involution, and transforming growth factor beta 1 and 2. These are these pleiotropic hormones that we know during normal lactation actually help the involution process, but actually their levels dramatically go up during breast cancer. So it's a confusing. Do they actually decrease risk by forming cells to shut down, or do they promote progression through having them grow? Well, here's the levels during beginning, middle, and wean samples, and the only one. So, for example, MMP2 goes down during weaning, which is actually I found very interesting, and MMP3 goes down, and MMP9. They all go down, even though the proteinases themselves go up in the extracellular environment. But TGF-beta 2 dramatically, significantly goes up during weaning. So again, this is telling me that this protein here is the key to what's going on during that weaning process. This is the thing we need to study to figure out, in other words, the question is, which pregnant and nursing women are going to get breast cancer? Which ones are not? And it's even harder. It's one thing to have a tumor and study it. We're trying to predict, in the absence of a tumor, who's going to get it. Right now in cancer, everybody's focusing on once a person has a tumor. Wouldn't it be better to know who's going to get it and prevent it or treat it early, as opposed to say, well, you've already got cancer, we're going to do surgery, give you chemotherapy, give you radiation, file you with surveillance, oh, by the way, it's $500,000. I'm not kidding, or at least 100. After you add it all up, more frequently. Or to say, we're going to prevent it and it costs you $10. So, I mean, but it's a lot harder, it's a lot higher bar to reach, because you can't accept toxicity, and most of those individuals you're screening never get cancer, so they lose interest and all the rest. So prevention strategies are definitely, for populations, the best way to go. They're the cheapest, they're the most logical, but they're the hardest to convince people of, because if you have cancer, you're like, oh my goodness, I'm going to die, I better get something done. If you say, well, you might get cancer, you want to do this, it's a lot harder to sell. That's why not everybody has their colonoscopy for colon cancer. Not all women have their mammogram for breast cancer. And not all people, get vaccinations for one reason or another against certain cancers. That's actually a debatable topic. But again, these are prevention strategies, but they are the most effective way to prevent cancer or to diagnose it early. So, again, remember that we were talking about exosomes communicating from one cell to another, and we want to look primarily at TGF-beta 2, and we want to make sure that these exosomes can get in breast cells. we want to strategize exosomes in different women based upon the amount of TGF-beta2 that the exosomes have. So we had women who had a lot of it and women who had a little of it. We wanted to look at things that drive cancer, like epithelial mesenchymal transition. Anybody know what that is? Anybody know what epithelial is?
Well, that's what's on top of the thing, right?
It's a shape. It's epithelial cells, they're lining cells in the body, but they're generally
Inside, not outside.
they're right, they're inside. But they're generally cuboidal in shape. Mesenchymal cells are more linear in shape. When we're born, prior to birth, as a fetus, there are 3 types of cells in our primitive developing body that form. Endoderm, mesoderm, and ectoderm. Remember that? Did you take that, Jack, in school? Anyway, the cells have different shapes and form different types of cells. But the point is that epithelial cells are cuboidal and they sit at rest. Mesenchymal cells are linear. They can wiggle through. Cancer cells become mesenchymal. Cancer cells that are mesenchymal metastasize and kill people. Cancer cells that are cuboidal look like normal cells and are far less likely to metastasize or kill people. So, that's why we want to look at EMT. These proteins are related to the development of EMT. Vimentin, alpha smooth muscle actin, and filamentous actin, because actin fibers are the cell membrane fibers that stretch. And so when they go up, it changes the shape of the cell. It pulls the cell apart. It's the kind of the construction project of the outside of that cell. So the levels of that are indicative of what's going on. Well, remember again, we wanted to first to see if exosomes can get inside cells. So, MCF7 cells, Michigan Cancer Foundation cells, they get inside cancer cells. This is a cancer cell. And these green dots, which is green fluorescent protein in the exosome, gets inside normal cells too. So that scared me, because that means that maybe these exosomes can be delivering messages to the normal breast. Remember, these are milk exosomes getting inside cancer cells and in normal cells to drive what's going to happen in that cell, both normal and cancer containing. Then we added exosomes that had a lot of TGF-beta high, to cancer cells to see if the cells grew slower or faster. They grew faster, significantly faster, with the exosomes versus control. That word is again. What does that mean? Cancer cells grow faster if you have this thing? Is that a good thing or a bad thing? It's a bad thing, right? It means that something bad's going on when you have too much of it. And then we added again TGF-beta2 exosomes. Here, okay, by the way, we talked about cuboidal and linear. So in normal epithelial cells, MCF7 cells, they're cuboidal and clumped together. When you add a little of the low exosome, TGF-beta2 exosome containing cells, they're pretty much the same. But when you add a high levels of TGF-beta2 exosome, you see how they put these little filopodia and buds? They have kind of like legs, and they can crawl, and they can wiggle, and they can metastasize. So basically, it's for making these cancer cells change shape and become more aggressive. We also had a neutralizing antibody. When you add high levels here, and then you add a neutralizing antibody, it goes back to the way it was, which again says this is what's happening. TGF-beta2 is what's causing these cells to change their activity. Anyway, you can see the kind of the legs that are forming. We call those filopodia, by the way. And here's yet another picture. Now, these are in normal cells, normal breast cells. This is the control. It's kind of hard to see, I apologize, but those are cuboidal cells touching each other. When you add a lot of TGF-beta 2, straight TGF-beta 2, just pure out of the chemistry bottle, look at the cuboidal shape. You see, I don't know if you can see how they're linear. Here they're a circle. Here they're more like a muscle cell. And these are the kinds of cells that can spread and get in blood vessels and in lymphatics and go elsewhere to another part of the body, like the brain or the lungs or somewhere you don't want them to go. If you have the low TGF-beta 2 containing milk exosomes, they're still cuboidal, just like the control. But if you have high levels, they become linear. See that? You see how their shape is far different. So, again, I think this is telling us that during weaning, high levels of TGF-beta 2 in a woman's breast milk may be a way to predict their risk of getting breast cancer. So, remember I talked to you about the proteins that correlate with these morphologic changes? So, alpha smooth muscle actin, FAC, filamentous actin, and vimentin are three proteins that are related to EMT. Higher levels when the cells change shape, more linear as opposed to cuboidal. And in fact, this picture is not very high, but basically the vimentin is there's more red when you have a lot of TGF-beta 2 in the cells. There's more filamentous actin, and there's more smooth muscle actin. So higher levels of actin are what cause the cell to change, or allow the cell actually to change shape. And they're higher when you add higher levels of exosomes containing high levels of TGF-beta 2. And again, some women have high levels, some women's breast milk has low levels. So, a few things to take from this. TGF-beta 2 is upregulated in breast milk exosomes during weaning early involution, when the breast is trying to deconstruct itself back to normal. Exosomes containing high levels of TGF-beta 2 cause changes in both normal and malignant breast cells, consistent with development and progression of breast cancer. So maybe there's a role for TGF-beta 2 to influence risk and maybe eventually screening women for levels of this in their breast milk may be a way to determine if they're going to get breast cancer during or shortly after nursing. So that's all I've got for you, but I'm happy to answer any questions. Thank you.
Questions for our speaker today?
You said the occurrence of breast cancer is increasing.
Uh-huh.
Is that just because there's more survival, and so the more genetics are there, or is it other external factors as well?
The numbers are higher each year in terms of incidence. That may well be simply because the population, I don't know if those numbers are, if we adjusted that for population increase, how much of an increase there would be. I would suspect it's a relationship of just more population in the United States as well as earlier detection. So whether there's, independent of that, there's also a true increase in incidence is not certain. The good news is that even though the number of cancers being diagnosed is going up, the number of deaths is staying the same, which means that the death rate, since there are more women in the United States, is actually going down. Not a lot, about 2% a year, but something.
You mentioned ultrasound was used to detect the tumors in nursing women. Can ultrasound be used to detect tumors in non-nursing nursing women? Because
Sure.
I know a lot of women don't want to do a mammogram because they feel it's painful and uncomfortable.
Yeah, so the technology for imaging the breast is becoming far more diverse. Used to be primarily mammography, and still that is the workhorse. Up until very recently, mammograms were done with an AP and a lateral, top down, left, right. And they had to compress the breast, which hurt. And so there's now what's called 3D mammography or tomosynthesis, which takes digital multiple images of the breast, which is actually more predictive of the presence of a tumor, especially in a woman with dense breasts, because it's not obscured by the fact that you've literally got one picture for a whole breast. You've got many pictures, and you can look at slices of the breast, and the compression is less. So the compression problem is not as bad with tomosynthesis or 3D mammography, but there is a slight amount of compression. There is still radiation, small amount of radiation, but not, the compression is not as bad. I mean, I'm told, I'm not, obviously, myself.
Yeah.
Yeah.
Are there any recommendations that could be derived from this research as to how the weaning process ought to occur? Would it be better to wean quickly or slowly or early or late?
No, I mean, I'm not aware that there's any difference. Essentially, what I think our data is suggesting is that during the process, either the growth of the breast in preparation for nursing, or the weaning process where you have to deconstruct all of that back to its normal pre-pregnant state, either that has gone awry or not. And we need, if there's a way for us to find out as early as possible which of those, in which women it's gone awry, the better. And to screen those women in some way. And I think that's what I would take away from
So
it.
it's purely diagnostic and there's no recommendation you can make for anything that can do different. It just
Well,
happens.
yeah, I mean, the only alternative is not to get pregnant or nurse. And frankly, the greatest risk is pregnancy itself. Because even if you don't nurse, the breast is going to enlarge during pregnancy and it still has to involute. I mean, in some ways, some people would argue that if you have the breast enlarged and then shut down on its own, the breast becomes engorged with milk and then has to somehow evacuate that milk if the baby's not taking it out. And that's probably not better either anyway. So lactation is a good thing. It's not a bad thing. It's pregnancy that really is the primary driver of the cancer. Whether or not you nurse, it still has to involute back to its normal pre-pregnant state. Yeah.
Since the breast is enlarging during that period, have you thought about looking into the hypoxia-inducible factor, the HIF?
Right. There's 40 and 70 and 95. Which one are you talking about?
Well, I have looked with the 90.
Okay. I haven't looked at HIF 90 or any of the other ones, but why do you think that would be especially
I'm
interesting?
thinking about the breast is enlarging, so that means there is more mass available, so that means the oxygen that's being present is decreasing, so therefore, who knows, maybe that factor might go up with the androgen.
It's certainly possible. I haven't looked at it. That doesn't mean it wouldn't be important. I honestly don't know. Sure. Yep.
You mentioned something about TGF-beta 2 antibody they're using in some of your experimentation.
Right.
Is that something that's has potential for use in women who have higher levels of TGF-beta 2?
Well, that's exactly what I'm suggesting might be useful. I mean, the truth is, we have to do a larger study to validate it before we would ever propose that. In at least a few hundred women, nursing women. And I mean, the hard part about this kind of work is, ultimately the FDA, the Food and Drug Administration, to recommend a test for use, wants the hard endpoint, cancer. And cancers in women of this age are not common. It's only at best maybe 11 or 15%. And and you have to wait 5 to 10 years. So it's a tough study to do. Doesn't mean it can't be done, but it's a large, it'll take a lot of women over at least 10 years period of time. So we have to convince some funding agencies to let us do that. Yep.
I noticed how you said breast cancer increases after like the first pregnancy. Well,
No, wait, no, sorry, I don't know that I said that.
maybe I got misunderstood.
Well, go ahead, what was the
Yeah,
question?
because my aunt had breast cancer in her first kid.
Uh-huh.
I guess he had a mental condition and he passed away. I want to know if maybe breast cancer and that could possibly be correlated.
So, what I hope I said was that the influence of pregnancy and lactation on breast cancer is related to the age of the first pregnancy.
Okay.
Age.
Okay.
Yep.
Does pregnancy have a large effect on other cancers related to women, like ovarian cancer?
Not that I'm aware of. Ovarian cancer, pregnancy, so birth control pills decrease ovarian cancer risk. It's about, so actually most ovarian cancer, the most common, this is off topic, but okay. The most common type of ovarian cancer is serous subtype ovarian cancer, that's just the pathologic appearance of the cells. And in fact, it doesn't form in the ovary. It actually comes from the fallopian tube. So the most common form of ovarian cancer is actually really ovarian tube cancer. And so what's happening, of course, is that the fallopian tube transports the egg from the ovary. It pops out of the ovary, goes into the fallopian tube, transports it from there into the uterus, and that whole process occasionally, goes awry. So birth control pills, by regulating, that process of the egg going into the tube and the rest of it, I know decreases risk. But whether pregnancy is, the same as like birth control, I don't know. That's a good question.
Okay, and with that question, we'll need to dismiss, but know that Dr. Sauter will be down here in the front if you have more questions, and also will be in the Joyce Room in the corner cafe. Let's thank Dr. Sauter one more time.